So hello everyone to this week's ESDR Kitchen live webinar. This week it's a sweet and sour with two absolute outstanding speakers or fighters in each corner, two absolute titans of biomarkers. We have the luck to have Christoph Schlapbach as a co-chair of the last minute. He agreed to co-chair this session with me, and he will introduce the first speaker of the two. So Christoph, thank you.
Yes, it's my great honor to introduce Kilian Eyerich to this sweet and sour session. He is the chair and professor at the Department of Dermatology and Venerology at the University of Freiburg, and he also holds a guest professorship at the Karolinska Institute in Stockholm. His main scientific interests are in the interaction of the adaptive immune system and resident epithelial cells in inflammatory skin disease. And his vision is nothing less than to revise basically how we speak and categorize inflammatory and autoimmune skin diseases. So he's really interested in doing research that is clinically meaningful and has a molecular basis. Kilian is a PI in numerous translational research projects and clinical trials. He has published more than 150 scientific articles in peer-reviewed journals such as the New England Journal of Medicine and Nature and Science Group type publication. His work received numerous awards, including European Research Council grants and a Heisenberg professorship. He serves as a board and advisor in numerous academic and pharmaceutical scientific organizations, and he is being currently the deputy editor of the JID, our mother journal. So it's really a great pleasure to have you, Kilian, and we're very much looking forward to your talk.
Thank you, Christoph. I'll make it a bit shorter for Catherine, so we still have time to have the discussion. Catherine is obviously, speaks for herself. She's professor of dermatology and therapeutics and a consult dermatologist in London at the St. John's Institute of Dermatology at King's College and at the Guy's and St. Thomas Hospital. She co-directs the skin therapy research unit and also leads the clinician in national hospitals. She, most importantly, in regard to today at least, she just led two major publications on biomarkers, what is the promise and what's the reality, and that's going to be the talk of today. Maybe on her, just very briefly also, she chaired the UK NICE guidelines on psoriasis and the British Association of Dermatologists guideline on biologic therapies. Shows she's really a titan in that field, and I'm really looking forward to the discussion of the two of you. I'm really looking forward and see if we get out of it any clearer. So it seems a bit of a mess right now. Thanks very much for having you here. I'm looking forward to your talk.
Thank you.
Thank you, Curdin. I mean, that's not really fair because you already gave us the result. And you should be really neutral, right? And my role is to be more optimistic. So let's start. Catherine, thanks a lot. I'm really looking forward to do this together with you. And thanks for letting me to be the optimistic side of the debate today. Just to very briefly introduce you where we are coming from, I think we have to make one thing clear, and that is that in this world of these highly heterogeneous diseases that we all do know and love as inflammatory skin diseases, and that's at the next slide, Catherine.
Yeah, can I just interrupt because oh, here we are.
Yeah, sure, thank you. So what we all know and what is possibly an advantage or a disadvantage, no matter the matter is how you see it, is that we have hundreds of different diagnoses. And they come from a time which is at least 150, if not 200 years old, and did not really change. And most of these diseases are not well known. There are no clear defined endpoints. The pathogenesis is not well studied. We do not have any approved drugs in them, and we still have to learn them, most of them at least, together with long names. And just as the next click, you see a brief overview about all these different diseases. The question is now, can we really leave it there? And the answer is clearly we cannot, because we clearly have a misfit between this descriptive nomenclature of inflammatory skin diseases at the one hand side and the techniques and the possibilities that we do have on the other hand side. We have growing therapies with a very specific mode of action, and now we have to fit them into the disease ontology, or the other way around. We have to fit the disease ontology into the new possibilities, molecular possibilities that we do have. And at the next slide, you do see what we actually need and want. Molecular diagnostics or modern diagnostics, image-based analysis. We will discuss what biomarkers actually are, and Catherine will give you the overview there in just a second. We need them to, at the top left side, predict the natural clinical course of a lot of diseases. Are they self-limited? Are they progressive? Are they maybe even resulting in inflammation and comorbidity in other organs, which we cannot predict to date? Which of the atopic dermatitis patients would develop an allergic asthma, or psoriasis patients would develop psoriatic arthritis? It's totally unknown with the tools that we have at hand today. But even the precise differential diagnosis is sometimes really difficult when it, for example, comes at erythroderma, scalp lesions, pustular lesions of the hand, and so on. We have clearly limited, but we need to know, because at the lower right side, you see the enormous advances that are made in the field of atopic dermatitis in blue or psoriasis in red, with all these different therapeutic approaches and therapies. And we do not really have a good idea at this step which patient would need what kind of therapy, what we can expect in terms of treatment response at an individual patient's level. So we do need these biomarkers. There's no need to debate that. And Catherine, I know you're the biggest supporter of this theory. So let's go to the debate. And I would really like to start being a bit optimistic at the next slide by giving you at least a little hint that there might be biomarkers that we could really use, which are already more or less known. And when it comes to theranostic biomarkers, we do not have any biomarkers that predict a therapeutic response at baseline yet. But we have, for example, interleukin 19, which can actually pretty quickly tell us whether therapy would be successful or not. What you see at the left side is in the solid line, the serum levels of interleukin 19 in psoriasis patients, and in the dotted line, you see the PASI response. And that pretty much shows you that IL-19 is very quick. And at the right-hand side, you can see that patients that do not get a PASI 25 response, so the non-responders, at week 2 do not have decreased levels of IL-19. But the PASI 100 responders at the right-hand side, they really have a reduction of IL-19. So IL-19 can be the predictor of the response of different therapies in psoriasis already at week 2 when it comes to readouts at week 16 or later. So it's not that bad, or is it, Catherine?
Thank you, Kilian. I will say at the beginning, I think we are agreed actually on the fundamental point that biomarkers are essential to better medicine, better healthcare for patients. I think my brief though is to a reality check. And what I'm going to cover in the next 8 minutes is definitions. The terminology around biomarkers is complex and confusing, and what we're trying to do, where we are in the research landscape, using the example of psoriasis, although the same is true right across medicine, and the biomarker development pipeline. In other words, what we need to do in order to reach that promise that Kilian is going to speak about shortly. So first of all, biomarker definitions, and both the FDA and the EMA have come up with helpful definitions around the term biomarker, which is short for biological marker. The FDA scope is wider because it includes physiological and radiological and histological characteristics, whereas the European Medicines Agency defines only biological molecules found in blood, fluids, and tissues. And in truth, that is where a lot of the research is focused on. And the type of biomarker, there are many that we're already using in clinical practice, clinical and imaging, but really the focus in the last 2 to 3 decades has been around molecular biomarkers. And that is because the perfect biomarker is on the causal pathway between the exposure of interest and outcome. And therefore, as Kilian indicates, by understanding the underlying molecular mechanisms in disease and response and drug and how drugs work, one can then identify perfect biomarkers, whereas imperfect biomarkers, in other words, those not on the causal pathway, will be more likely to be subject to confounding and other factors influencing the final outcome. And therefore, that's where all the focus of attention has been. And latterly, of course, digital biomarkers have come into play to enrich the clinical information and longitudinal outcome measurements such as heart rate and so on. So this is an area of massive research, but fundamentally, these are the 4 areas that we want these biomarkers to help us. In other words, identifying disease susceptibility, better diagnostics, as Kilian has identified, prognostics, in other words, how bad will the disease become, and then predictive, can you predict whether a treatment is going to work or not? And the ideal is that by introducing a biomarker into the clinical decision making, or indeed in scoping out interventions for population health, you will therefore, with the biomarker, as compared to no biomarker, improve understanding of disease or risk of disease, make behavior change, so knowing one's risk might lead to healthier behaviors or interventions, or to aid better clinical decision. And I think that's one key thing where we know to date in inflammatory skin disease, the evidence that biomarkers can influence and improve health outcomes. That is where the big gap in our current evidence base is. And as we all know, there are no biomarkers routinely recommended in 2 of the most common inflammatory skin diseases. And as Curdin mentioned, we, as part of this Biomap consortium, which is a Europe-wide public-private consortium aimed at identifying biomarkers' importance in atopic dermatitis and psoriasis. As part of this, we have scoped out the landscape of biomarker research in psoriasis and also in atopic dermatitis, but I'm going to talk about the psoriasis reviews, and this was done in collaboration with the IPC. And we focus on disease progression, severity, and comorbidity. These are important outcomes. And this is one reality check, focusing on the scientific community on honing down what are the outcomes that are really critical, where should we be focusing our attention? And then systemic treatment response. Can we identify biomarkers that make it more effective and safe? And this is what we found. So we had a very wide, sensitive search strategy. So it was very inclusive. And as you can see, we used the same search strategy for both response and progression, but then separated them out. But in the end of the day, we identified only 7 candidate biomarkers that might be, and this is a big question mark even, but that where there was some evidence that these candidates may be worthwhile taking forward for further research. And in the disease progression, in related primarily to disease severity and psoriatic arthritis, 22 candidates. And these were the ones listed here. And I won't go through the detail of this, but just to make a few points. First of all, the research is dominated by genomic and proteomic biomarkers. Secondly, that it's a very, in general, until very recently, a candidate-driven approach. In other words, you can see there HLA-C0602, known as the major disease susceptibility locus in psoriasis, and that has been tested as a potential biomarker in a lot of research. And the third point is that, and related to that, that many of these biomarkers relate to known disease biology in psoriasis. But what it also brought out, and these themes are common themes, common to the literature in psoriasis, but actually common to biomarker literature in general. There is an important research gap between discovery and evidence for clinical benefit. We have scientists producing huge numbers of novel biomarkers, but not nearly enough efforts and link up with industry, with diagnostics, with evidence generation of benefit to health outcomes. In other words, very limited external validation. Effect sizes are often statistically, but may not clinically relevant. A second theme is the clinical utility of biomarkers changes over time. So as healthcare changes, exemplified in psoriasis by the step change in effectiveness of the new tranche of biologic therapies, the P19s as an example, do we really need a biomarker for TNF response? And this is a real challenge in biomarker medicine more generally. And the third is the study method limitations. I've listed them there, but part of this is driven by researchers using data sets and samples that are available as opposed to the ones that are purposely, prospectively collected to address a specific study question. This is the biomarker development pipeline. And this is a third reality check. This is what we need to do in order to make these biomarkers a reality. And the discovery validation pipeline through to so-called qualification. That is approval by EMA and FDA that this biomarker is valid and will answer a specific clinical or add to clinical care in a specific context of use. And the point here is it's long. So as I've said, the discovery pipeline is very active as evidenced by our literature search. But what's lacking is assessment of analytical validity, clinical validity, and clinical utility. And one example, this is a study that I was involved in led by Paola Di Melio in the UK, where she has identified this predictive biomarker of adalimumab response, NF-kappa B signaling in type 2 dendritic cells. And this pipeline here, just illustrated schematically, illustrates what work has taken and done, and it took nearly 4 to 5 years in the end. She has identified, discovered this biomarker, she's replicated it, she has refined it, she's validated it using a different technology, and then clinically validated it in a small external cohort. However, that cohort was only 14 patients. So in order to bring this through, we need to scale up and develop an assay that can be used in clinical practice. The assays that we use to discover are not fit for purpose for routine clinical use, and we need to analytic validity, clinical validity in cohorts beyond our own, and then does it actually make a difference to health outcomes? And this is all to do, illustrating that the challenge in biomarkers. And so there ends my talk. And the last bit, of course, is bringing it into clinical practice. I know Kilian may mention this. I'm actually not so pessimistic because collaboration such as Biomark, where you're bringing all the key stakeholders together in order to drive this critical pipeline through to the clinic, I am reasonably optimistic. Thank you. Kilian.
Thanks, Catherine, that you ended this in an optimistic way, because in between I got a bit depressed, I have to admit. But now let's really focus on, in the last minutes, what is the promise that we have with biomarker response? We have the unmet need, we have all the obstacles that we discussed, and now basically I want to touch 4 points very briefly. Why are we not there yet? First, the markers don't exist. We would absolutely reject this possibility, right? We are scientists and optimists, okay, so that doesn't exist. Second, the data sets don't exist that we need. Third, the methods don't exist that we need. And 4, and that is what you have just heard, the translation is super difficult to really make a biomarker enter the daily clinical routine. So when it comes to the methods, I just want to give you 2 examples that we have the methods in place. And the answer to a lot of this is artificial intelligence that you can hear everywhere. And these are just two brief examples of what machine learning can do. This is from 2 clinical trials, has been published recently. In these 2 clinical trials, AD patients have been asked every day, how much is your skin bothering you and what kind of medication do you need? And then the severity was assessed. And here the colleagues tested a machine learning algorithm to predict the severity of the skin at the next day based on these 2 variables. And at the right-hand side, you see these black lines. These are 4 patients and their severity scores over time. And you see in blue what the algorithm predicted. And especially the patient at the lower right side, for example, had 1 flare that you can see. And the algorithm quite well predicted that. Now you can ask, of course, is it useful and clinically valid to predict the severity of the next day? And the answer is probably not. But this just shows you, with just these 2 variables, you can create something like an algorithm that predicts skin severity using machine learning. And you can, of course, do that more complex. At the next slide, you see these QSPs that are now everywhere, which is quantitative system pharmacology models. And that basically gives a neuronal network, and at the next slide, please, Catherine, the neuronal network, all information that you have in a complex system, biological system, for example, such as an inflammatory skin disease. This is a QSP coming from the academic world for atopic dermatitis. And for example, you put in here keratinocyte experiments treated with IL-4 and IL-13, or clinical trial data using dupilumab, or mouse models of atopic dermatitis. You just put in all data pieces you can get, and then you try and identify the whole complexity of the system and how you could interfere with this. It actually comes from the weather forecast, these kind of models. And at the right-hand side here, you see that this model from the left side was used to predict the easy response to given drugs. In a small population, again, of dupilumab non-responders, it was predicted that drug 1, which is in this case fezakinumab, an anti-interleukin 22 antibody, would result in a better outcome. And again, in the black dots here, you see the true easy scores, and in the colored lines, you see what the algorithm predicted. So that means that these neuronal networks, machine learning algorithms, and biostatistical, bioinformatical tools, they are basically in place. We have them. What we do need is now the data. And there it comes back to what Catherine has said. The next slide, please. We just need to collect whatever we have. And Biomap is a very good example, I think, for the private, public, academic, and pharmaceutical world partnership. But of course, we do have big competitors here like Google Health and IBM Watson that just collect every piece of data that all of us in the community always publish, right? So now the question is this data set really enough, or do we need another layer of granularity? And there, I'm personally convinced that we actually do need better metadata. And this is something, Catherine, that we already observe also in Biomap. It's not enough to give a diagnosis and maybe the age or the gender. We really need more granular metadata of the molecular high impact or high throughput data that we do have. And at the next slide, you see just one example of an initiative that we started at the Karolinska Institute in Stockholm and that we mirror now in Freiburg, where we take full body images of the patients at different time points. So we do not take the easy score or scored score, but we rather have 400 gigabyte of image data of each patient. We combine that with patient-related outcome measurements and different kind of omics, which would be lipidomics, microbiome, transcriptome, proteome. And then we do observe that over time and after certain therapies. And we aim here at including more than 5,000 patients in order to really get the whole spectrum, heterogeneous spectrum of inflammatory skin diseases that we have been discussing before, and then to really generate a new disease ontology based on molecular mechanisms. And here, I think this should be an open initiative, and everybody that would be interested in collaborating contributing some patients is more than welcome. The final question is at the next slide, how do we translate then if we really have these biomarkers? And here you see the number of apps. One more, please, Catherine. That is all that is image related, can be directly analyzed by applications on our smartphone or on the laptop or wherever. And we have now 243 apps which are used in inflammatory skin diseases currently. Most of them work like a diary, but basically they collect information. And as soon as they collect information of what could have been the triggers and a selfie snapshot and medication, we might be able to generate better data and to give better predictions in the future. The second thing is the molecular, and that is the more really biomarkers at my last slide. How do we translate molecular information into daily clinical practice? Because most of these tests are really heterogeneous. They are difficult. The primers used need to be the same. The temperature needs to be the same. The methods, the materials need to be exactly the same. And people need to be very experienced. It's lengthy, it's costly. So we need fully automated systems and labs on the chip. And this is my personal conflict of interest because I'm one of the co-founders of Dermagnostics here. But what that basically is, you see this disc in the middle, and you put in some kind of skin sample, which could be a paraffin embedded skin. It could be a mini biopsy. It could also be tape strip or serum. And then you have just by centrifugation and heating, you can basically get single cell suspensions, isolate RNA, do a real-time PCR, and do this biocomputational analysis of the molecular data you receive in order to get the full answer within something like 90 to 120 minutes at the maximum. There are also, and you see that down there, a lot of other companies that work on similar solutions. In other words, we might be there that we soon have something like an application selfie of a smartphone of the patient together with, at the entrance, a tape strip and molecular information that will be generated fully automated in these lab on a chip systems in reasonable time and at pretty low costs. And then we are really there that we can say we implement biomarkers into daily clinical use. And so that's the promise that we do make. And it's not years away because some of these products are already available on the market. And also Dermagnostics is very close to have that first inflammatory skin disease biomarker pair on the market now. And with initiatives such as Biomap and better metadata, bigger data sets, we will pretty soon be there that we have better biomarkers that we can validate. I'm fully optimistic there. I stop here. Thanks a lot, Catherine. It was really fun. We might have some minutes for discussion.
Shall I stop sharing?
Thank you very much, Kilian and Catherine, for these excellent and brilliant talks. I'm sure there are questions. Unfortunately, I forgot actually in the beginning to mention that the attendees can write in their questions into the chat and we'll transfer them to the two speakers. Luckily, Nick Reynolds is a old dog in that regard and knows how to do it. So he already has the first question. Thanks to two of you, this is an interesting presentation. And I think it's a very valid question he has. So why is there a mismatch between cancer and inflammatory skin diseases in terms of these biomarkers in development already in clinical practice? I think both of you can reply to that question.
Kilian, do you want to answer? Because I think it relates to your focus on molecular diagnostics of biomarker discovery, which is probably relevant.
Yeah, no, I'm happy to give at least, I think it's two explanations. One is the honest one and one is the one that I would like to give. The one I would like to give is that inflammatory skin diseases have one big difference compared to cancer, and that is they are relapsing remitting. So it's not one usually progressive, not monogenetic, but driven by a few driver mutations disease that is progressive like cancer, but it can be better and worse. The heterogeneity can be higher. The fourth dimension is more difficult to assess and we only get a snapshot. So it's more difficult to assess biomarkers in the field. But the true answer is, I don't believe that. The true answer is, I think there's much more money and interest in the field of cancer research, which is why they started 10 years earlier, which is why they are now more advanced. And in inflammatory skin diseases, we just need to catch up. And I think we do at the moment, and we are in a good way here.
Yeah, I mean, I think I'd agree with that. Of course, with cancer, you have the perfect internal control. You have the person with the cancer, and then their internal control. I think that simplifies it. And also, I agree. The other thing that's driven biomarkers is drug development. So it's a sort of synergistic thing. And so with more targeted therapy, you're able to unpick and identify new targets, if you like, and new biomarkers. If you look at all the new drugs on the market, virtually all of them have got co-diagnostics. So drug discovery does drive, but actually in terms of the questions and identifying those at risk, I think that's the other thing. I mean, there's a lot of biomarkers in cancer medicine around diagnostics, but not so much in terms of implementation about population screening, for example, of cancer risk. So it illustrates the differences. It depends on what biomarker you're after as well. You know, what your biomarker is trying to do.
Related question has just been dropped into the chat, and the question is by Marcella. How much does biological variability affect the promise of molecular biomarkers in terms of clinical validity? Maybe Catherine, you can answer.
Yeah, it's a good question. And I think also, I mean, I think it's interesting, isn't it? If you take psoriasis, which is a clinically a variable, not perhaps as variable as atopic dermatitis. Nevertheless, we have a drug, the P19 class, that achieves a clearance in a very large proportion of patients. So even though you've got clinical heterogeneity and biological variability, you've still got a drug that works in most patients that's targeting that particular pathway. So I think it probably depends on what the biomarker you're going after, you know, what the outcome you're trying to have a biomarker measurement, as to how much that variability might be important, and then also the disease that you're dealing with. You know, there is a sense that there's more heterogeneity within AD, for example, than there is in psoriasis. So
maybe personal question, follow up on that, on both of you. And Kilian already showed his new technique. Do you think there will be a single biomarker that will be used in the clinic in the future, or is it going to be a mixture of different biomarkers, obviously with the difficulty of the utility in daily practice?
I mean, in the literature review, as you will have seen, there were biomarkers that were common across disease, more severe disease, psoriatic arthritis, and treatment response. So, but I don't know the answer to that question. But I think that comes back to actually a more fundamental question that, as a community, we should explore and decide what area should we be really focusing our research and discovery efforts on. In other words, which area in clinical practice or a population or an individual do we really most need the biomarkers, actually, and which are going to have longevity? You see, for me, I think identifying early on which people in the population are going to go on and get more severe disease and more comorbid disease at a population level, that's got much more promise. In terms of drug efficacy, actually, you could argue, well, you know, you can try a drug for 3 months, and then if it doesn't work, just move on to the next. So it's an interesting question, but I think it's where we probably, you know, in COVID, we were all focused on the same question: how incredibly effective was that in terms of discovery science? I think we might be spread too thin, but I don't know what Kilian thinks about that.
I think that it will always be a mix of biomarkers personally, and of course we want to have the ideal overview, and biomarkers might be also at different levels. I would agree that genetic biomarkers probably at an individual patient are not useful, but proteomic, transcriptomic, maybe epigenetics, as we see here in the chat, right? These are highly interesting biomarkers. It will be a combination. I personally think that it would rather be a combination of 40 to 50 biomarkers and not 60,000. But for sure not one, Curdin. I think we would agree on this.
There's another question by Lynn. It's actually a fairly practical question, not so difficult to answer. Is there a centralized resource for sharing protein biomarkers similar to the GWAS catalog, or to Protein Atlas probably? Is there?
Well, there's a Protein Atlas, as you say, isn't there? But I don't know. I don't know protein. Actually, I don't know the answer to that question.
Does Protein Atlas have biomarker? Yeah, well, there's a little bit for cancer, which is, but again, that's cancer and not inflammatory disease.
I mean, one of the within Biomap, and I know Hippocrates and other consortia, there's sharing, you know, wide sharing and discussions about how to make these bioresources and information or data widely available, the sustainability, because it's a very, very good point more generally about research to redundancy, and you know, open access data sharing is critical actually to driving more effective, quicker outcomes, I think. So
you would need a totally new level of harmonization, right? Because the biomarkers, I mean, you have screened them, Catherine. It's totally different models, totally different systems, and you cannot just compare all of them in one list, right? You need to establish standard criteria, and I'm not aware that something like this exists, although it would be a very important initiative, actually.
Yeah, it's a good question.
I mean, at the end of the day, there's this famous saying, BS in, BS out. No matter how elaborate your algorithm is, if you feed the algorithm with shaky and bad data, then nothing good will come of it. And yeah, I think there's maybe I can have a quick personal follow-up question to Kathy. What is your suggestion? What should we be focusing on as a community? What's your top priority?
Well,
I'd be curious to know.
Well, I mean, it's a clinical perspective, of course, but my sense is that we, and certainly in psoriasis, you know, we have very good treatments now. We have really very effective treatments, but we still see people with psoriasis suffering a life trajectory of untreated disease. And so, and also this association between disease severity and comorbid burden. So I personally would, and it's a common condition where most people have mild or less troublesome, less impactful disease. So my feeling is that identifying those at risk of more severe disease or more comorbidities early is where we could really make the biggest impact in terms of people's lives. And it's what people want to know. It is what they want to know. And also from a cost effectiveness point of view, that, I mean, maybe Kilian, your technique is going to be very cheap and easily accessible, but you know, one of the other big barriers to treatment effectiveness is drug cost. And I'm not convinced that biomarkers are actually going to reduce that much, in actual fact. You know, I think the margins, and drugs will get better and better anyway. They have in psoriasis, for example. So I think getting the right population to the right treatment as early as possible, I think that's where I would put effectiveness.
To calculate once, you know, what you mentioned before, the trial and error that we used to do already 20 years ago. What is the cost for the community actually that we could gain by not having trial and error? I mean, psoriasis, yeah, we're at 95%, but AD we're at 50 maybe, but even in psoriasis, it would be interesting actually to calculate what would be the gain that you can achieve by actually going 99%, right?
I mean, as one example, we just did a health economic analysis of using real world data of trying of the sequences of TNF antagonists versus ustekinumab and secukinumab, because that was where the data was. And whatever way you did it, pretty much, it was always cheaper to start off with the TNF antagonist with a biosimilar. You know, drug cost drives so much of the cost effectiveness models, although they're limited too, because they don't take into the social impact. That's the other limitation. But cost is important. Although it's not prioritized by the community. Kilian, in your paper recently, the cost of biomarkers was a low priority and interest, but it will be the thing that stops us using it in the clinic.
Yeah, but Catherine, now you get pessimistic again because you're a psoriasis expert, right? But let's really try and think about above the horizon. I mean, prurigo pigmentosa, PRP, right? There are these trained diseases we don't understand. So for me, a top priority would be if the biomarker can predict it's chronic or not, is it septic or not, and what kind of signature does it have? So what kind of therapy can we give to the patient? And then ideally the next step would be antigen specificity, right? That would offer us positive therapeutic approaches. And I think we can arrive there. And I think that most of the inflammatory skin diseases which are chronic are actually autoimmune diseases, but we need to develop these kind of biomarkers and concepts, obviously.
I mean, I think your point about those other diseases, yeah, we've talked about mainly AD and psoriasis. So I do agree that the molecular drivers, the biology of all of this, 2000 dermatology diagnoses, that I think understanding and having better molecular diagnostics around that would be, yeah. Well, you definitely are an optimist, Kilian.
Shouldn't we researchers all be optimists? Otherwise, it's
No, it's true. It's true.
It's a little difficult, the business we're in. So there's a final question that has not been addressed so far. Maybe we can make this the last one for today. Sheila asks, my understanding is that molecular biomarkers predict average risk at the population/cohort level, but I wonder what are your thoughts on the potential for individual risk prediction in inflammatory disease? The polygenic risk score community are very dubious about this at present, I think.
Well, I can only fully agree, right? What we have at the moment is basically nothing that we can use at an individual patient's level. There are now initial biomarkers, and IL-13 is one of these examples, right? That you can really use at an individual patient's level to predict or differential diagnosis. But we are at the very beginning, and especially the biomarkers Catherine identified in this huge work and effort of the meta-analysis, like HLA-CW6, I mean, they are not absolutely useless at an individual patient's level. It's just, we need to do proof of concepts. We need to refine our work to get better and better. And we can do that by getting better data sets, and big consortia that work on combining clinical information with the molecular data sets. I think we are on the way, but absolutely yes, at the moment we are not there.
So thanks very much, and I think it's a great, final statement to close the session for today. Maybe what Lynn already wrote in the chat as well. To thank you to two of you. It was a great presentation and very thought provocative. And at least I'm also a little bit more optimistic for the future. Even not much, but a little bit. And just not to forget all of you to register for the ESDR this year, the 51st. One in Amsterdam in September. Next ESDR Kitchen, maybe we'll quickly go back, is on Spidion, thanks a lot, on the building of network meta-analysis. So that's going to be in 2 weeks time. And we have a new session at ESDR this year. I was a bit too fast. So there's going to be an oral presentation in late breaking concurrent session. So we have a new deadline for late breaking abstracts. It's going to be end of August. So please submit these as well if you have any great new data to submit there. So thank you very much. Thanks to two of you. Thanks, Christoph, and have a great afternoon. Bye-bye, everyone.